All right, you guys, we're going to start with chapter 8, lower invertebrates. Okay, so we just did the higher invertebrates. And I just want to make sure that there was no confusion. Those 4 characteristics were for the characteristics of chordates, so of vertebrates. So that was a connection between the higher invertebrates going to vertebrates. So again, those 4 characteristics are dorsal hollow nerve cord, notochord, pharyngeal gill slits, and post-anal tail. So I just want to clarify that in case there was any confusion at the end of the third video for chapter 9. That was a long one. So for chapter 8, it's 52 slides. So we're going to do 26 slides on the first half and then 26 slides in the second half. All right? A lot of key concepts here. Feel free to go back and look at them on your PowerPoint. So first question is, what are the definitions, what are characteristics, of an animal? All right, animals have to be multicellular. They have to be eukaryotic. Remember, characteristics of a eukaryotic organism is that they have a nucleus, they have membrane-bound organelles. That means mitochondria, the rough and smooth ER, vacuoles, all right, lysosomes. That's what we're talking about here. No chloroplasts, remember, because chloroplasts, that is for plants. We're talking about animals, all right? We're talking about their DNA is linear, well, our DNA is linear, right? These are all characteristics of eukaryotes, specifically animals, okay? They cannot produce their own food, so they are heterotrophs. Add that word in right there. Oh, I really like that orange. Heterotrophs, all right? They can actively move, with the exception of some sessile adult forms. Sessile, non-moving. Okay, they're stationary. Sessile, stationary. They stay in one spot. They stay there. You get 2 types of animals. Invertebrates, what we've been talking about in chapter 9 and now chapter 8. Most marine animals really are invertebrates. And then, of course, are vertebrates, which is everything we have been doing before chapter 9, or after, technically. Chapters 10, 11, and 12. All right? Okay. Sponges. Phylum Porifera. You're going to need to know some of these scientific names. Definitely Phylum Porifera. So we've got a lot of awesome specimens. If you remember that day that we did a kind of exploratory lab, all right? All these cool specimens. The sponges, this one's my absolute favorite. I love this one, right? Totally cool. And hopefully you guys did do as I asked that day and you got them wet. Right now they feel real stiff and not nice to touch. When you get them wet, natural sponges, right? And then you feel them against you. They're so soft, so nice. Ooh. Okay? And they're great at being exfoliant because there's something called spicules in them. I don't know if you remember when we did that exploratory lab, looking at all the different types of things about marine biology. There's those triangular shaped spikes. That's why I can describe them. And that's why sponges are actually great at exfoliating, getting dead skin off. Okay? So sponges, basic shape, very, very simple, asymmetrical. That means you cannot cut them and get 2 perfect halves. No matter what you do, no matter how you do it, you're not going to get 2 perfect halves. So they are asymmetrical. Very simplistic form, very, very early evolutionary branching. Sessile, permanently attached to the solid surface. Non-moving, remember, sessile, stationary, stay in place. The water flow, apologies. So you would think that the water would flow, let me see if I've got a good specimen here. The water would flow in through this opening, apologies, and then out through. But it actually does the opposite. It comes in through the sides, and it comes out through this main opening right here, all right? I've got another good specimen. Give me 1 second. Let me find it for you. I did one slide yesterday, and I was like, oh, it's not that great, but that was what I wanted to do. And if I don't find it in the next 5 seconds, yes, I found it. Here we go. Oh, it's as if you're right back in classroom, right? So you see this one right here? See that beautiful opening right there? All right, so the water comes in through here, and how does the water get in there? Because they have these special collar cells right here, and this is a flagellum that sticks out, and they literally siphon the water in through all these itty bitty openings right there. See right there? And then you can see inside those openings, and so they push the water in through these little openings on the side of the sponge, and then it goes out through this main opening and it pulls out. I think we watched a video that shows it with the dye, if I remember right, a while back. If we didn't, let me shoot me a message and I'll gladly find it, put it on there. So anyway, so it goes through the ostia to the spongiocele and now the osculum. This is called the osculum, this opening right up here. There's no tissue to them. The collar cells are the ones who do all this. The flagellum is constantly moving and provides the current. The pinacocytes, these are layers of cells, outer covering. And then you've got the archaeocytes, amoeba-like and can move through the body. These are stem cells. They can become anything, whatever the body needs it for it to become. Spicules, those are those triangular shaped that I just described, that I drew on the board just a few minutes ago. Cell type, a skeletal element to support the sponge body, and it's made of spongin. Calcium carbonate again, silica, or protein spongin, depending on the species. Look at groups, sorry. This is something if you want to do, I forgot about this. This is something if you want to do at home. If you got Play-Doh, you got a little sibling, or you got some Play-Doh, you might want to do it and actually build yourself a little sponge. And literally grab some toothpicks and poke the holes and really visualize the sponge. If you want to take a picture of it and put it, send it to me on Remind, or you know what, don't do that. I'll put it on the discussion group. I think, yeah, that'll be fun. Let's have some fun, you guys, okay? Don't have to do this, this is just for fun. I tell you what, you do it, it'll count for a sticker point. Deal? How's that? We'll see who's paying attention to the video. Ooh. Okay, see, you got this really nice picture of this. So you've got 3 body forms. And it's just showing you different styles. And that's why I had all these different samples for y'all to look at. This is the most complex. That's this one right here. That's the leuconoid. Highest degree of infolding, many chambers, largest sponge. Osculum. That's my little one I just had. Right? Very simple. Asconoid simplest. Tubular small. And then this would be more of your cyconoid. Sort of-ish. I don't have a great cyconoid, honestly. I think that's the last one sample I need to get for y'all. But don't need to know, it's not red. It's just interesting. Look at that. There we go. Those are just beautiful. I just love sponges. I think they're very cool. Some sponges are just ginormous. You're going to see a picture coming up in just a minute. They're filter feeders, suspension feeders. Thanks to the collar cells breathing in, they'll grab everything out of the water. And their reproduction is asexual, simple budding. So they'll fragment a piece off and send it on through. They can also do sexual reproduction. Most are hermaphroditic. Eggs from the archaeocytes and sperm from the modified collar cells. Depending on the water temperature or photoperiod, so basically based on the amount of light and darkness in the 24-hour period, will lead to gamete production. So when it's just the right temperature or the right day, time of the year, they'll go ahead and release the gametes. Their larval stage is a planktonic amphiblastula. That's what it looks like. So blastula, remember, is a ball of cells. So amphiblastula is the same thing in sponges. Blastula is what it is in humans, animals. Amphiblastula is the same thing, just different name, slightly different setup. Here's the life cycle for sponges. Really kind of neat to see it. Ecological roles of sponges, why are they important? There's competition. Corals and bryozoans, these are moss animals. They all need a hard substrate. So sponges are in competition with corals in order to go ahead and have enough space on the coral reefs. There's also predatory-prey relationships. Very few species eat sponges because those spicules, it's not agreeable for organisms to eat them, but some animals can eat still sponges. Some sponges also have a chemical deterrent. So if you eat them, it's actually not, it'll make them sick. And you're toxic, but the hawksbill sea turtle. they're like, bring it on. They can eat them, no problem. So they'll come up and chomp away right at the sponges. They got no problems eating them. It's kind of cool. You've got symbiotic relationships as well. Some are mutualistic hosts. Some species have 2 times the amount of bacterial cells for every sponge. Or commensalistic. Organisms will live within the canals. Some have 16,000 snapping shrimp in 1 loggerhead sponge. It's really crazy. It's amazing. They're also very important in nutrient cycling. The boring sponges are important in recycling of calcium. They're burrowed to the corals and mollusk shells. And obviously they're extremely important for commercial uses. Class Desmopodia, these guys have been harvested. Whoopsie! Ah! Have been harvested like crazy. I've got the dropsies today. Have been harvested like crazy. They grow. You tear off a piece, they'll grow again. They'll grow, just harvest them. They'll hold 35 times its weight in liquid. It'll take 5 years to reach maturity. And due to regenerative properties, fishermen will reattach fragments to a rock, throw them back in, get some more out, and it's nothing better than this to polish metal. A lot of them have also been used for cancer fighting because it's going to block the actual copying of the DNA of cancer cells, specifically the carrion species, and there's questions as to whether or not sponges are actually antibacterial. Kind of important right now. All right. Cnidarians. Oh, we've been talking about those guys. Remember, the sea is silent. Cnidarians. This includes your jellyfish, your hydroids, your corals, your sea anemones. Cnidocytes. These are the stinging cells for prey capturing and for protection. These have radial symmetry. So if you think of a flower pot, you can cut a flower pot from the top any which way and you still have 2 halves, right? That's radial symmetry. Same with a sea anemone. You can cut it multiple different ways and you'll have 2 perfect halves, right? Radial symmetry. Boom. Radius. You can do a radius. You can go across. Boom, 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 boom. Sponge, there's no symmetry to it whatsoever. If I try and get 2 perfect halves, it is not gonna happen. No story about it. That's no symmetry. This is radial symmetry. Now, bilateral symmetry is where there is only one way to cut to get 2 perfect halves. If I cut this beetle this way, I have 2 halves, but are they symmetrical? No, they're not, right? You're gonna have a head and a bottom. You're not gonna have 2 perfect halves. There's only one way to cut. Same thing with a human, right? There's only one way to cut down the middle to get 2 perfect halves. No, we're not including internal organs here. Nah, just externally, only one way to cut down to get 2 perfect halves, right? But cnidarians have radial symmetry. We okay with the difference in symmetry? Cool. When we do dissections, I'll talk about that too. All right? Body forms. Cnidarians have 2 types of body forms. They have the polyp, and then they have the medusa. The polyp, like a coral polyp, is where the base is at the bottom, and the tentacles are at the top, right? Just like a coral, just like a sea anemone, right? No problem. Medusa is more of the jellyfish, where the tentacles are at the bottom and the dome is at the top. What's interesting is the word for jellyfish in French is méduse because of that body form. So instead of an A, it's an E, méduse. So polyp benthic form with cylindrical body with opening at one end surrounded by tentacles at the top. This is the only form found in anemones and corals. Medusa, free floating stage, that's also known as a jellyfish, right? Here we go, showing that radial symmetry again, and then showing the polyp and the medusa forms. Cnida, this is your stinging organelle. They have nematocysts. All right, this is really, really cool. If you've ever been stung by a jellyfish, and you have wondered why in the world it is so painful, you're going to get an up close and personal look at what hurt. And it's this right here. This is the nematocyst that went into you, and these are the barbels that locked it into your skin. Okay? One of the things you've probably heard is to take sand. It was actually my students who've been lifeguards who told me about this. You take the sand and you scrub it onto the jellyfish thing. And I thought about it and I was like, what? And then I was like, oh, it makes sense, because what you're doing is you're sanding down the epidermal skin layers to get rid of that and get it down to the layer where these are no longer embedded in your skin, they're gonna fall out, okay? The other thing you can use is vinegar. Yes, there's the commonly said thing to use to pee on it. We're gonna go back to that one. You don't wanna do that one, by the way. And then there's also meat tenderizer. Now, what do meat tenderizer, urine, so don't pee on it, please, and vinegar all have in common? I'm gonna give you one answer, maybe a hint. Proteins. What do you think we're doing to this? Denaturing the proteins, because we are changing, I'm gonna say it again, vinegar, urine, and meat tenderizer. Meat tenderizer deals with salt, high salt content, and then these two deal with pH. You're denaturing the protein by either changing the pH or changing the salt concentration. So you're denaturing the protein, and this no longer is working, all right? The nematocysts are the spearing type of cyto, which is located within a capsule in the cell, and this will shoot through, embed itself in your skin, and it'll do that as well in any kind of prey that the cnidaria are trying to eat. So it's very interesting, very painful, but very interesting to understand the mechanism behind it. I have more slides on this one, and then I will stop. To say that they are a dangerous species is an understatement. It can cause, depending on what the type of jellyfish is that stings you, it can cause immediate intense pain, burning, weakness, nausea, headaches, back abdominal muscle spasms, and in some cases, it can kill you. Portuguese man of war, we definitely have had these in Florida, right here in our beaches. This is a very pretty, colorful, pinky purple jellyfish. What they do is they will inflate the sail so that the wind will catch it and actually push them around. Their tentacles can be tens of feet long, so beneath the water surface. Dead or broken tentacles can still sting you. Vinegar and meat tenderizer are great to use. Box jellyfish, these are the ones that are found in Australia. They'll kill you within minutes. You do not use vinegar with box jellyfish. The box jellyfish, for some reason, evolutionary adaptation, these guys will actually, when vinegar is exposed to the nematocyst, it will continue to sting you, causing these reactions to be worse, and it's why it can cause death even faster. There is one jellyfish in Florida that is very, very bad, Invertebrates jellyfish. It will lead to hospitalization. These are jellyfish scars, jellyfish sting scars. They can be pretty bad, you guys. It's definitely something to worry about. I've seen jellyfish that have gone through broken pillars and get broken up, and that happened to my mom and I. We were swimming, my brother was scuba diving, and we were just getting stung. We didn't know what was going on. And then the guy on the boat was like, it was in Mexico, and he was saying Medusa, Medusa, and in French we knew that Medusa was jellyfish. We realized it was jellyfish, and we all got out of the water, and it wasn't. We were like, man, there's no jellyfish, and we realized it was the tentacles that had been blended up in the boat propellers, and they were stinging us like crazy. Luckily, it was nothing dangerous, but it was uncomfortable. I mean, the jellyfish thing, when you don't know what's going on, it's definitely painful. It's like, it can be like a bee sting, and if you get several jellyfish tentacles hitting you at once, it's like a bunch of jellyfish, a bunch of bee stings at once. It can be painful, but even if it's not a dangerous species. Hydrozoans, class Hydrozoa, mostly colonial. Their colonial form contains two types of polyps, the feeding polyp and the reproductive polyp. Hydrocorals secrete a calcareous skeleton. Most will produce floating colonies. Portuguese man-o-war is an example of this. So Portuguese man-o-war belongs to the class Hydrozoa. Scyphozoans, that's your true jellyfish, like your moon jellies, right? Very, very gorgeous, beautiful specimens. The Medusa is a predominant stage. They've got photoreceptor sense organs that allows them to determine whether it's dark or light. They'll stay at depth during the bright sun and during the at night, and then they'll kind of move up and down through the water column. It's really neat. I love the jellyfish. They're so pretty. The box jellyfish. They are not very big, but they have very long tentacles. You see that definite, distinct cube shape, hence the name class Cubozoa. Cube shape to them. Very strong swimmers with complex eyes, not eyes as we think of as our eyes, but eyes where they can, those photoreceptors so they can sense the sunlight. Very tropical, voracious predators, and they primarily feed on fish. So the fish get stung down here in the tentacles, and then they slowly move them up into the bell, and that's what we call the bell up here, and that's where they get eaten. And then our anthozoa. These are your sea anemones. They are obviously very benthic. Gastrovascular cavity is divided. They have an opening right here. That's their mouth, right? So if we feed them, just like you guys do with a coral feed, right? We feed them here. Some of the corals you guys feed are actually anemones. We feed them. They close up, and they're going to later poop out through that same opening. All right? I'm going to stop the video here, and we'll do part 2. Okay?